Welcome everyone. So this is number 26 of the Instruct-ERIC Structure Meets Function webinar series. This is a slightly different one today. So this is coming from two of our centres in France and in Spain, who are going to be giving introduction to some of the new technologies that they have in the centres. Just a brief introduction to Instruct for anybody that is new to the webinar series. Instruct is a research infrastructure spanning across Europe, with 16 now member countries and organizations across Europe. We very recently, just last week, added Greece and Slovenia to our members, and researchers from all of these countries and organizations can receive funded access to the services that are on offer at our centres. Services in structural biology, including sample preparation, biomolecular analysis, and some 3D structural analysis as well. So yeah, these are services that are available to everybody worldwide, but funded access is available to our members. So some of these services are going to be described to you today. So these are new services. So slightly later on, we're going to hear from the team at Instruct France 1 at IGBMC. But first, we're going to hear from the team at in Madrid at CNB-CSIC and the Imaging Centre I2PC as well. So to kick us off, we're going to hear from Javier Conesa. So they're going to share their screen now, and yeah, the floor should be yours.
Well, good morning, everybody, and thanks for the introduction. So I'm Javier Conesa, and I'm going to present today this talk entitled Cryocorrelative Light and Electron Microscopy at the National Center for Biotechnology. The correlative microscopy lab with this new service that we are offering through Instruct is integrated within the cryo-EM facility at CNB. That is an Instruct center located in Madrid. Here are the members of the service from left to right. Durand, head of the service, Francisco Javier Chichón, María Teresa Bueno, Noelia Zamarreño, David Delgado, Jonathan Puchirilo, and myself. So in the cryocorrelative microscopy lab, we have implemented mainly two workflows. One is devoted for organelle level information, organelle level resolution, and is cryo-FIB-SEM tomography, and the other one is for going to near atomic resolution by subtomography through cryo-electron tomography, and we are using the FIB-SEM for the lamella preparation. Regarding the sample preparation, we have implemented two protocols, and we are using one or the other depending on the thickness of the sample. When we are talking about around a thickness no larger than 10 microns, we are using plunge freezing, and that includes cell monolayers grown on top of the grids or attached to them, or purified samples. And for that, we are doing plunge freezing in liquid ethane cooled down by liquid nitrogen. And for that, we are using these two equipments. One is the Leica EM GP2 that is more devoted for cell preparation. And then we have also the Thermo Fisher Vitaboard Mark IV. Also to notice in this step is that we are using correlative fiducials that are bits of 1 micron diameter that are visible with an iron oxide core and covered by a biocompatible layer that is emitting autofluorescence. So it's they are fiducial that we can follow in between the different imaging modalities. For thicker samples, from 10 microns up to 200 microns, we are doing the vitrification by high pressure freezing, using different supports, including 3 and 6 millimeter planchettes, and also we have implemented the waffle method. The first step after the sample preparation is the fluo imaging, and this step is common to both workflows I introduced at the beginning. So for the cryofluorescence imaging, we are using two microscopes. One is a wide field isoscope A1 microscope, and we are using this one to check the sample quality and also to collect 2D information from our samples. And we are using this one when when we have a frequent event or a yes or no event, like for example, when a cell is infected or not infected by a virus. When we need a more precise localization, it means 3D coordinates, we are using this confocal microscope, an LSM 900, equipped with an iris-scan 2 detector. And in both microscopes, we are using for keeping the sample at the liquid nitrogen temperature, the Linkham stage that is automatic, is connected to the microscope computers, and we can operate it from the microscope itself. Here an example of grid with the cells attached to the surface. In this case, T lymphocytes staying with 2 different fluorophores that you can see in more detail here in this 3D confocal structure. So here we have in red, we have mitotracker signal. In green, we have a specific endocytic compartment with a protein label with GFP. And also in gray values, you have the reflected light that is also giving us information about the sample quality preservation. These rings, these concentric rings, are telling us that the sample has volume, and that the absence of these rings in the area surrounding the cell is telling us that there is not much ice there. So after the vitrification and the cryofluorescence microscopy, next step is loading the samples in our FIPSEM microscope. We are using a Crossbeam 550 that is able to operate from 0.5 to 30 kilovolts. That is equipped with a FEXM that is able to produce high quality images, with gallium FIP, also with the software SenConnect that allow us to transfer 2D coordinates from the fluorescence microscope to the FIPSEM microscope. And also the microscope is equipped with a Leica cryostage. So for the first workflow, the one that is aiming at organelle level resolution, and the serial sectioning or the cryo-FIPSEM tomography. After imaging by the doing the cryofluorescence microscopy of our samples, then we load them in the FIPSEM microscope. First thing would be to take an image of the surface of our sample. Then we switch off the SEM, switch on the FIP, and we mill away a thin layer of the cell, as thin as 10 nanometers. That is our axial resolution. And then we repeat the process. We image again with the SEM the new surface, and we repeat and repeat and repeat. And at the end, we cover when we cover the whole volume of the cell, we finish with a stack of SEM images that we can align, reprocess, and finally render the 3-dimensional volume of the cell. And from there, we can study the distribution of the organs. Here, 2 examples of how the grids looks under the SEM imaging. Here is a virtual section of the cryo-FIPSEM tomogram that can give you an idea of the resolution that we can achieve. So we can identify several organelles, including mitochondria and their inner structures. We can locate the centrioles, the Golgi apparatus, endoplasmic reticulum, nuclear pores, different endocytic compartments, lipid droplets. So again, at the level of resolution at the level of cellular organ, around 10 nanometer resolution. Then we can overlay the fluo information with the SEM volumes that we have. And for example, in this case, it's allowing us to identify a specific endocytic compartment. We can distinguish in between vesicles with a positive or not positive signal. So the other workflow, the one going through cryo-electron tomography, we are using the cryo-FIPSEM microscope to prepare lamellas to make the sample compatible with the transmission electron microscope. So the thickness of the cell is around 10 microns. So if we want to get a high resolution information, we have to go through transmission electron microscopy. The sample is too thick to be analyzed. So in order to make it thinner in a gentle way, we do that using the cryo-FIPSEM microscope. So again, we have the fluo information, then we transfer it to the FIPS microscope. We prepare our lamella that is compatible now with the transmission electron microscope. We load it in our transmission electron microscope. We acquire the the TIL series. Then we reconstruct the tomograms, and then from these tomograms you can extract the repetitive structures to increase the resolution of your volumes through subtomoral aberration. So here, Thermo Fisher video that is simplifying how the lamella preparation is working. So we know that our event of interest is in this area. From the FIPS perspective, we position the milling patterns, and we remove away the material that we are not interested in this specific case, leaving just the area of interest within our lamella, now compatible with the transmission electron microscope. So here, to simplify the workflow, we relocate the cells that we previously analyzed by fluorescent cryofluorescence microscopy. Then, using the fiducial markers, we are transferring the coordinates of the fluo images to the FIP and SEM perspective, selecting the corresponding same bits in both imaging modalities. And once we transfer the coordinates of the fiducials, we say, okay, our event of interest is located in this specific point, fluorescence point, that is corresponding to this specific region in the FIP-SEM images. So we place here our milling patterns, leaving our area of interest within the lamella. Here an example of the lamellas that we prepare, here from the FIP perspective, and here with the SEM microscope high resolution image showing what cellular structure remains within the lamella. So once we have prepared the lamellas, then we transfer them to our 10 microscope, Geo Cryo Arm 300, to perform the acquisition of the TIL series. And then here to summarize the whole workflow, transmitted light here the section of our confocal stack. From the field perspective, the very same cell that is over there, and then we select the area that we are interested in. We mill away the areas we are not interested in. Then we transfer it to the TEM microscope, and here's how the lamella is looking in the TEM. And then we acquire the TIL series and reconstruct the tomogram. And you can see that we can identify several cellular compartments here. For example, mitochondria, microtubules, polyribosomes, coated vesicles, and that could be all. And thanks to all the people involved in this the implementation of this infrastructure, and also thank you all for your attention. I'm happy to take questions.
Thanks a lot, Javier, for this very nice and clear presentation. Is there any quick question from the audience at that stage? I mean, maybe I will just ask a very pragmatic access question. So in general, how long is a visit to the CLEM facility?
I mean, the whole pipeline could take around 1 week. If we start from the cells, not vitrified cells, up to the final volumes, it's taking around 1 week. But also it's important to consider that the cells need to be ready to be attached or grown on top of the grids before the vitrification. So it cannot be done in the home lab and has to be done here. The experiments could take longer than a week because you have to grow the cells and so on.
Okay, thanks. So we have a question from Rafael Lero. What would be the different option for access? Full pipeline? Can it be step by step? Can you say a bit more about that?
I mean, I think that we are flexible with that, and the whole procedure is accessible. But if users have already prepared the samples, we can incorporate them in whatever point of the pipeline.
Okay, thanks. And we have a question from Durand. So do you screen multiple lamella before choosing the one to study in detail? Transcription factors are promoting a new machine, no, TF, sorry, Darren, I do not know what TF are, a new machine architects that promotes this approach. Thermo Fisher, sorry, Darren.
I didn't get the last part of the question.
Yeah, so Thermo Fisher are promoting a new machine called Arctics that promotes this approach.
Yes.
So are you following such approach of screening multiple lamella before choosing the one that you are studying?
Actually, no. We want to save ice contamination. So once the lamellas are prepared, we are not loading them again in the cryofluorescence microscope because we don't have a cryofluorescence microscope online in the cryofixem. But I mean, we can modify the pipeline. We can adapt to the user. So if the user want to, after preparing the lamellas, load them in the cryofluorescence microscope, I mean, we are able to.
Thanks. We have a new question from Bruno Claros. Do you see issues with non-amorphous ice using the EM ice instrument? Tested lamella by diffraction, calculate Fourier transform?
Actually, for the high pressure freezing experiments, we just measure them by cryofixem volume imaging. We didn't prepare lamellas out of high pressure frozen samples and measure them in the cryo-electron microscope.
Thanks a lot, Javier. Any other question from the audience? No. Well, if you have any other question, you can ask them in the Q&A and Javier can answer online via the chat. So we'll move on to the next speaker. Thanks, Javier. So it will be David Herreros that will talk about the Flexi Hub.
I'll share my screen. Can you see my screen now? So, well, first, thank you so much for inviting me to give this talk in the Instruct webinar series. In this presentation, I will be talking about the Scipion Flexibility Hub, which is a new framework inside the Scipion. And the idea is trying to simplify as much as possible the design and definition of the workflows, but focusing on the flexibility part of the analysis. So before going into the Flexibility Hub, I would like to do a very brief introduction and talk about the reason why we decided to develop this Flexibility Hub. So as we all know, in cryo-EM, there are probably two main types of different heterogeneity analysis. The first one is the classical one, is discrete, which can be obtained using a 3D classification. And at the end, we are getting n number of different states. Usually it's not a very large number, but it can give us a good idea of what are the different stable conformations our proteins explore. And then we have kind of a new way of analyzing flexibility, which is more advanced, which is the continuous. And here the idea is that we are getting a much larger number of states. And we can not only see the discrete ones, but also we can infer probably some transition or the way the protein is going from state A to state B, for example, as you can see here in the video from this COVID spike. But I think the real question is if we really need to go into the continuous case, right? Because in principle, if we get enough number of discrete states, we might have probably a good enough approximation to the different conformation of the states, and that should be enough, right? By going into continuous heterogeneity. Well, for that, let's continue with this example of the COVID spike, and let's suppose we have a data set, we do some 3D classification, and we can reach, for example, these two different states, the 1-up conformation of the spike and the 3-down. And if we put this into a conformational landscape, let's say, at the end, what we are reaching is just 2 points, which, well, is giving us an idea. But let's compare it to the continuous case. And if we superimpose this, now we are seeing that actually we were missing a key structural information, and it was not just a few states, but actually it was a lot of things. The transition between these two states, but also a lot of different conformations that are living within each of these different classes, let's say. So actually going into continuous heterogeneity is work. And I would say that now we are shifting towards not doing just classical 3D classification, but going into advanced flexibility analysis. And for that we are seeing that there are many different programs right now coming out. It's a very hot topic. These programs can be divided into two different approaches. I will not comment a lot about these two different approaches, just saying that, of course, as any other program, some approaches have strengths, have weaknesses. And some of the main representatives, I would say, from this new advanced flexibility analysis could be the CNB-3D, 3D Flex, normal mode, Cryo-Dragon, Gaussian mixtures, and of course, many other programs that are coming out. But I think we are always having the same problem. And the problem is that really having this arrangement of the programs in a list is not very useful. I mean, at the end, if we are saying that a program has a strength or has a weakness, it basically means that for my dataset, for example, if I try Cryo-Dragon, it might work very well. But maybe if I try CERNIC or 3D Flex, the results might not be as good. But I don't have a way to really take these programs, run them all together, even do some combined analysis and say, okay, I like a lot the results from Cryo-Dragon, or Cryo-Dragon is telling me one thing, but then the CERN is other thing that is also interesting. So I think what we really want is to arrange the programs in a workflow, something similar to this, right? So we have some input particles and input data, and we want to have an easy way to run all of these programs in parallel, and then say, okay, I have these programs, and now I need tools to really analyze what is going on, going into the landscapes in an interactive way, get all the different conformations, understand what these programs are trying to approximate, even having tools for doing consensus and trying to combine this landscape into something more meaningful. And this is why we decided to develop the Flexibility Hub, because we were having the feeling that we are having a lot of software coming out, very powerful, but we don't really have a way to use all of them together to get the best out of our samples, the best out of our images and our proteins. So this is why we decided to work with the Flexibility Hub. And as I said at the very beginning, the Flexibility Hub is just kind of a section inside the Cypion. And the idea is that this section is completely focused on flexibility analysis, and is not only providing software to doing this or extracting these conformational landscapes, but also is providing you with tools that you can use to interactively explore the landscape and understand better what is going on with all these different states and conformations. So probably the Flexibility Hub will have kind of two pillars. First one, of course, is the integration. We need to provide or we need to give software. And right now we are offering CERNIC-3D, which is our own developments, but also Cryo-Dragon, normal modes. And our idea is to add more software like 3D Flex, like Gaussian mixtures, manifold EM, and so on. But integration by itself is not useful. So we also need to provide with interaction. We need to also to give ways to analyze all these different results. And for that, we are also developing interactive tools, which are allowing you to, in real time, go to any point in the landscape and actually see all these different states, possible transitions. If you have the states that are probably more stable or they are less represented by your particles, also tools for doing consensus. So instead of having just, for example, one run of the CERNICs and one run of Cryo-Dragon, try to combine them and to keep only really those states that you can trust, because the two programs have estimated a similar state for those, and all these kind of tools. So as a summary, what are we offering in the Flexibility Hub? Well, of course, we are also offering software. Right now, as I said, Cryo-Dragon, CERNIC-3D, also tools that are more focused on the structural model side, like Prodi and Continuous Flex, and more to come. We are offering tools for analysis, either interactive, that can be, you can work with them in 2D or 3D. I will show you some examples later. Also doing automatic analysis to more or less infer what are the possible best number of states that you can expect from your conformational landscape. Also, we are offering a new consensus tool, which in principle should allow you to combine all these different measurements into a single one and do a stability analysis and keep only those particles that are very stable. We are also providing ways to get and extract these conformational states from the landscape. The usual way will be doing refinements, but also we are providing the own way each different software has to decode or generate these different conformational states. And we are offering also extra utilities. These extra utilities, for example, will allow you to view your map in 3D, but also with some arrows painted, and these arrows are telling you which different regions of the map are moving to, so you get a better idea of what is going on with the motions of your protein. Also, this program that we are offering called SART, which is taking advantage of these conformational heterogeneity estimations, not to provide you with new states, but to correct for the heterogeneity and trying to improve the resolution of very, very flexible areas in your molecule and so on. So now let's try to see some examples of the flexibility hub, how it works, what we are trying to propose with this session as Scipion. So here you have a Scipion workflow from the flexibility hub, and you can already see more or less how we can start connecting different softwares, right? Like, for example, these blue boxes in the top are all the different estimations from different softwares, like the Cernix, CryoDragon, and so on, which are already giving us ways to explore, to get a landscape and explore it. But instead of that, we can try to go, for example, into the Flex consensus, which are these boxes in green, and this will allow us to extract only those particles that are really estimated in a similar way by the two softwares and exclude all those other particles that probably are garbage or they are not estimated well by many other possible reasons. And then you can go into the analysis part, which are all these pink or kind of red or well, this weird color here. And the idea is that with this, you can explore your landscapes. You can get all these different conformations. You can do refinements, or you can correct heterogeneity with SART. So you have many, many different options. And the good thing that this is already living in the Scipion environment. So you can take any of this information you are getting, and you can do a standard Scipion workflows like SPA, in the future probably also TOMO, and you can extract as much information as you can from this flexibility analysis. As I said, one very important part of the Flexibility Hub is not only providing you with programs, but giving you tools to understand what is going on with the conformational landscape. And for that, we are offering many different interactive tools. This one is one of the examples working in 3D, where you have your landscape in a three-dimensional way represented. So you can rotate it, you can place any point in your landscape to get in real time the conformation, and you can visualize it here. And even you have ways to connect from these tools into Chimera-X to do morphings or any other kind of advanced visualization, so you can understand better what are the different views that you can see in your conformational landscape. And then also we are working on not only 3D tools, but also 2D versions of them, which are giving you an extra piece of information, which is the number of particles that are appearing or that are placed on a given region of your landscape, which probably means that some regions are more stable than others, because you are getting a much larger number of particles representing that very specific state. So by combining all these different tools, you can extract more or less in real time the information of your landscape. You can annotate it, place labels, and more or less have an idea of what is going on. I think the other very important part is the consensus. This is a thing we are missing these tools that are saying, okay, you have an estimation of, for example, the Cernix, or even you have 2 or 3 of them, or estimations from different softwares. But the question is, can I really trust what the software is telling me? And of course, there are parts of the estimation that you can trust a lot, but some others might have a lot of errors due to the noisy conditions in cryo-EM, or probably because that particle is not really a particle, it's some kind of garbage. So the idea of this Flex consensus is to take a lot of runs of different softwares, can be the same software run twice or 3 times, or even different algorithms, and to have a way to put them in a common space and do an stability analysis. So now in an interactive way, you can keep only those particles that are stable, and you can do a subset into a Scipion and continue with your analysis and continue getting your conformations and so on. And then, as I said, we have also extra tools, which are not really to explore the landscape, but can also help in the understanding of what is going on with one state or with one motion. For example, these tools here where you can see the map or a structural model and place arrows telling you where different regions of the map are trying to move. And this might help in the understanding of the different states that you are exploring in your landscape, or even ways to go directly to structural models. Most programs nowadays allow you to get these new conformations at the level of cryo-EM density maps. But I think going into the atomic world is the next step, because once you are here, you can, for example, do normal mode analysis or any other kind of structural model-based analysis that is giving you much more information of the different motions, what is going on with your different states. But up to here, I have been talking about Flexibility Hub as a section in a Scipion, providing you software, tools, and way to do workflows, but also we are offering it as an extra service. And the idea is that you can send us your particles, your information that you have extracted from your analysis with Scipion or any other software. And the idea is that we will do this kind of flexibility analysis, trying to extract as much information as possible. And all this information about the service is in the Instruct webpage. And you have here all my colleagues that are mostly working on this new Flexibility Hub service, like Blanca, Roberto, and Marcos. And I would like to finish, of course, thanking the biocomputing unit, because thanks to them, we have all this Flexibility Hub, all this Scipion, and so on. Of course, all the funding, which, of course, without the funding, it would be impossible to develop all these things. And that's all. Thank you so much for your attention. And if you have any questions, I will be very happy to answer.
Thanks a lot, David. Great talk. We have a question from Dimitri that first says that it's a very nice presentation, but he would also like to understand, so to know if he understood properly. So his question is, do I understand correctly that I need only the particles for flexibility analysis, no original micrographs?
Yeah, so micrographs are not needed. It really depends on the kind of analysis you want to do. We have, I think the most standardized way to do this kind of flexibility analysis is at the particle level. So you will need in principle a set of particles. Of course, it's not just a big set of particles. You need to have CTF information, alignments, and so on. And with that, you can enter into all these flexibility analysis by itself. But it's also true that you can work with other types of data. Like, for example, if you have different conformations extracted from 3D classification, there are also ways to move them into the continuous flexibility world, also with the structural models. So in principle, there are different ways to enter into this kind of analysis, but it will be always in a late, in a later stage of a cryo-EM workflow. So when you already have particles with alignments, different classifications, and so on. Yeah.
Okay, and we have a second question. What requirements does Flexibility Hub need? For example, a large box, about 512 pixel size particle. Sorry, I do not understand well how the consensus works. Is there a way to validate which of the different approaches or solutions are better? Are they weight or calculated an error?
Yeah. So for the first question, the box size, well, in principle, there is no limit on how large box size would be when you try to do flexibility analysis. But I would say most programs will impose downsampling to your particles. Like, for example, if you run CryoDragon or the Cernix, most likely they will ask you to downsample internally the box size to, for example, 120 pixels. The idea is that first you are getting performance, but also since all the particles have a lot of noise, you probably won't be able to extract very, very fine conformational changes because you cannot really trust them. So the downsample is somehow safe, but this is an option that you can control, and it would also depend on your setup, right? If you have a better GPU or you have more or less memory and so on. But in principle, there is no limitation in this sense. And then for the consensus, yeah, I have gone very fast over it, but the idea is that you provide as an input different estimations from different programs or even the same one several times. And what you are getting is an error measurement for each of these different algorithms. Of course, these error measurements are going to be related to each other, right? Because what we are trying to do is to move all the programs into the same space. But the idea is that once you have run this, we are able to split again the errors and tell you, okay, so if you want to continue working with CryoDragon, for example, these are the errors that we have gotten for the CryoDragon particles. And you can now keep only those particles in CryoDragon that were consistent also with the other softwares. Or you can take, for example, the Cernix particles and do the similar kind of analysis. So in principle, it is not like they are combined into something and you don't have a way to go back and recover the previous information, but it's just going into something common internally. And then you have also a way to split the errors and analyze each of the different runs independently.
Thanks a lot, David. So as we are running a bit late, we will move on with the next presentations. So thanks to both of you, and we will move for the presentation from Instruct France 1, where we have Alexandre Durand and Catherine Birck talking.
So I will start.
You will start, Catherine?
Yes.
Very good.
So good morning. So today I will present the new BLI and FIDA biophysics technology at Instruct-ERIC France 1. So users have access to these technologies through Instruct services. As an introduction, I will briefly present the activity at Instruct-ERIC France 1. So this activity ranges from sample preparation to 3D structure determination. In sample preparation, we produce protein in bacteria, insect cells, mammalian cells, and perform protein purification. We also perform biophysical characterization to check the homogeneity, the stability, and the affinity of the biomolecules. And we determine 3D structure using electron microscopy, crystallography, NMR, and super resolution microscopy. At Instruct Center France 1, users have access to a toolbox of biophysical methods for quality control and for functional characterization. For quality control, light scattering, DLS and SECMALS, differential scanning fluorimetry, nano-DSF, and small angle X-ray scattering, SACS. For functional characterization, we have microcalorimetry, thermophoresis, analytical ultracentrifugation in sedimentation velocity and sedimentation equilibrium mode. And the last two I will present today, the flow-induced dispersion analysis, FIDA, and the biolayer interferometry, BLI. So FIDA is a capillary method employing Taylor dispersion analysis for measuring the hydrodynamic radius. So here is a picture of the equipment with a fluorescence detector on the right side. On the top, you will have the 1 meter capillary. And below the capillary, you will have the sample trays. In this case, samples are loaded in vials, but you can also load the samples in plate. As it is illustrated here, the radial diffusion of the sample in the capillary under laminar flow will lead to a symmetric Gaussian distribution. This FIDA peak or Taylorsgram will be analyzed in the software. Two experimental parameters, the sigma square, which is the width of the peak at the half 8, and the residence time, which is the time, the measuring time where the peak reach the detector, the top of the peak reach the detector. This parameter will be used to determine the sedimentation coefficient and also the hydrodynamic radius, which is inversely related to the diffusion coefficient using the Stokes law. So the FIDA application are the characterization of biomolecule and the binding events. So you can use FIDA for quality control to check the size of your sample, the presence and number of aggregate, the presence of free fluorophore. I will come back to this later. And you can also quantify protein in fermentation media. You can also use FIDA for complex formation to check protein-protein, protein nucleic acid, and protein-peptide interaction. And you can also use FIDA for liquid-liquid phase and exosome analysis. So to perform FIDA measurement, you need a fluorescent biomolecule, which is called indicator in this case. So you can buy a dye-labeled DNA, RNA, or a peptide, so with FITC or FAM labeling. You can also use a GFP fusion protein, or you can use commercial labeling kit to label your target protein. In this case, you will need to purify your labeling protein to remove the free dye from the label protein using a G25 colon, as it is shown in this picture. So you can then measure the label protein with FIDA, and using the software, you will see in this case that in the sample, you have 89% of your protein, but also you are contaminated by 11% of the free dye. So FIDA provides an explicit measurement of respective fraction of free versus conjugated fluorophore. In the FIDA software, you can also use, you can also predict the hydrodynamic radius from PDB data. As it is shown here, the structure of an homodimer So you can calculate the hydrodynamic radius. So it's in this case 3.4 nanometer. From the homodimer, you can extract the monomer structure and also calculate the hydrodynamic radius of the monomer, around 3 nanometer in this case. And then you can measure the protein in solution using the FIDA and compare the measured hydrodynamic radius to the predicted one. In this case, you see that you get 3 nanometers. So the protein in solution at this concentration is a monomeric form. For the data analysis, you will fit your data with a Gaussian function. So you can use a single species model or a multiple species model. You see in this case that the single model in green doesn't fit very well the data, but the multiple species model in orange fits the data. You will also get the number of aggregates, 11 in this case. So it means 275,000 aggregates per milliliter for this sample. For titration, if you perform a titration series for interaction studies, data points are fitted to the FIDA binding isotherm to determine the KD and also the size of the complex. This is an example of a FITC peptide protein interaction that was measured at Instruct France 1 by Judith Hose. And you see that in the curve of the hydrodynamic radius as a function of the analyte concentration, fitting this curve, you will get the size of the complex, 3 nanometer, and the KD, 0.68 micromolar. So the advantages of FIDA is that you will obtain a structural parameter, the hydrodynamic radius. You can study molecular interaction and get the hydrodynamic radius of the complex. This is a simple and rapid method with very low sample consumption. You can analyze LLPS. You can detect and count large aggregates. This method has a high matrix tolerance, so you can perform the measurement in cell lysate and all types of buffer. And the system has full automation. The limitation, it requires labeling with a fluorophore. You can only detect complex formation if the delta RH is upper than 5%, and you can face sometimes non-specific binding to surfaces. So the second method is BLI interferometry, biolayer interferometry, which is an optical technology for measuring biomolecular interaction. So first, you will need to bind one partner to a biosensor at the surface of the biosensor, and then you will dip this biosensor in a solution with the second partner. And if you have an interaction, you will this will cause a shift in the interference pattern, as it is shown here. And this wavelength shift measures the change of thickness of the biological layer here. So you can measure in real time the interaction. So you will get binding specificity, the rate of association and dissociation, and or you can measure concentration with precision and accuracy. At Instruct France 1, we are equipped with a new generation of BLI instrument, the Octet R8, and we have different types of biosensor to perform the experiment. For example, we have the NENTI to bind the histag protein. So this is a typical binding kinetic experiment using BLI instrument. So first, you will dip your biosensor. Here, it's a streptavidin biosensor in buffer to get a baseline. Then you will load the biosensor in the solution with the first partner that will be attached to the biosensor. Here, it's a strep tag protein. Then you will make another baseline. And then you will dip the biosensor in the solution of the second partner to measure the association, and then in buffer to measure the dissociation. So these are the raw data that you collect at these different steps. So here the first baseline, the loading of the first molecule, the baseline, the association step, and the dissociation at different concentration. So to perform an experiment, you will need to prepare a plate with the different samples. In the first column, you will put some buffer. In the second column, you will put the protein that you want to immobilize on the biosensor. In the third column, you will put buffer again. And in the fourth column, you will load a different concentration of the second partner. And then you will have to write a small program with a different step before to start the run. So the advantages of the BLI is it is a label-free detection system. So you will get time-resolved quantification of interaction. So you will determine the affinity and the kinetic parameters of your system. It is a simple and rapid technology with low sample consumption. So we are equipped with the 8-channel system, so it's a medium throughput. And you can measure sample in complex sample matrices. And this is also full automation. The limitations are that you have to immobilize one of the binding partner, so it's not in free solution. Sometimes you have to face it's challenging we have problem with non-specific binding. And another thing is that the molecular weight detection is upper to 150 Dalton. So it's not applicable to some small molecule applications. To finish, I would like to highlight the paper from Kenneth Verstraeters that was published last month in Nature Structural and Molecular Biology. In this paper, the authors elucidated the structure and mechanism of leptin-mediated LEP-R assemblies, and especially they reveal that the leptin LEP-R heterodimer form trimers in solution, and not 2-2 or 2-4 assemblies, as it was previously suggested. So we contributed to this paper through an Instruct access, and we determined the KD of the 1-to-1 to the 3-to-3 interaction using sedimentation velocity experiment. So the trimeric assembly of the heterodimer was illustrated on the cover of the journal by 3 avocados. So thank you for your attention. I will be happy to take questions.
Thanks a lot, Catherine, for this great presentation. So we have a few questions coming up. We'll definitely take the first 2 ones, but as we are running a bit late, you might have to answer the other ones in the chat. So the first one is about the FIDA instrument. What is your experience with elongated non-globular proteins, Catherine?
Alors, elongated, I don't know, but I have some experience with unfolding protein. I don't know if the user, and so in case of unfolding protein, you will measure very higher hydrodynamic radius than you expect for the protein of the same size.
Thanks. I guess the user can ask more questions in the chat if needed.
Sure.
So we have a question from Frank as well. Did you have any protein sticking to the capillary for the FIDA experiment? And how do you get rid of it?
Yes, of course. It's when you always work with surfaces, you have problem of sticking. And in FIDA, it's sometimes the case also, but you have different capillaries, standard capillary and coated capillary, and now a new generation of coated capillary. So you have, it's less and less sticky in some case, and you can also use detergent and BSA to decrease the sticking, and it works fine.
Okay, great. For questions of time, so there's still question in a Q&A. Could you, Catherine, reply to it in the Q&A directly, writing the answer if it's possible?
Sure.
I think we need to move on. So Alexandre, if you can maybe start with your presentation, that will be the last of today, about the chameleon sample preparation technique.
Yeah, can take over. Okay, so hello everybody. So I will present you the chameleon, which is available on the cryo-EM facility of Instruct Center France 1. The chameleon is a device to improve sample preparation for cryo-EM. Catherine quickly introduces the Instruct Center France 1. I will not go into the details of it, but all the equipment that I will speak about are accessible via Instruct or other programs. So if you ever want to make running projects with us, you can contact me. We offer the access to a complete set of equipment to run cryo-EM projects, either by single particle analysis or by tomography. At the heart of the facility, of course, we have a transmission electron microscope. We're equipped with a glacios that is mainly used for screening, but also for data acquisition, thanks to the K2 direct electron detector. We also have two Titan cryos, including a very recent G4 Titan, equipped with all the recent development and technical advances available, producing very high image quality for high resolution imaging. We also offer access to sample preparation method and equipment, such as the VitroBot, of course, the chameleon, which is the topic of today's presentation, and also for particularly difficult samples, we can also offer access to streptavidin affinity grids. And on the edge of sample preparation and imaging, we also have Orega FIB scanning electron microscope that can be used either for imaging or for making lamellas for tomography on the transmission electron microscope. So we offer access or power service for the entire workflow for cryo-electron microscopy. This is a simplified view by Thermo Fisher of what the workflow looks like. It's very linear, so we have all the equipment to perform all these steps. Unfortunately, life is not as easy as this, and most of the time, things do not go smoothly, and you have to come back to the previous step based on the result of what you have done. And as you can see on this graph, actually, many of the raw converge on a single step, which is the vitrification, and I think this is because it's still the bottleneck for many projects in cryo-EM. So VitroBot for years has been the standard for freezing grids for electron microscopy. And the aim using the VitroBot is to obtain a good grid where you have high particle density for single particle analysis. You want good contrast, mainly thin ice. You want to have a perfect particle distribution, of course, low noise in the background, and very importantly, you want to preserve the structural integrity of your object you are studying. And if everything goes well, we will all end up with this kind of grids which are just perfect for imaging. Fortunately, most of the time, or very often still, we do end up with empty grids or grids where we have clearly dissociation of the object, or we have aggregation of the sample, which does not really bear very well the vitrification step. These problems have been well described and understood and are linked to the protein denaturation at the air-water interface. There are many papers describing it now. And so just to make a long story short, you all know that these samples have a tendency to accumulate at the air-water interface when you freeze the grids with the VitroBot, many sample preparation method actually. And this can have two major consequences. Either that you have preferential orientation when your sample just stick to the air-water interface in a preferential interface. And this is detrimental for 3D reconstruction afterwards. Or you can also have protein denaturation, where the parts in touch with the air-water interface just denaturate. There are several limits to the traditional blocks and plunge freezing approach as it is done by the VitroBot. The air-water interface is, as I just described, is of course a major problem. It's probably due to the relatively long incubation that happens on the grid due to the principle of the method. There is also the need to screen the grids to assess its quality and to control what you have done before going to data acquisition. And very often, or at least still happen, that we end up with completely black grids or broken grids that cannot be used, and you waste a lot of time at screening a microscope. Also, even though it's not requiring a lot of a sample in terms of volume, still what you deposit on the grid is wasted mostly in the blotting paper. It would be good to have a way to improve that, especially for difficult samples which are hard or costly to produce. Finally, the result can be user-dependent. Prove that there is a learning curve to be able to handle the grid safely. In particular, for occasional user or new user, a large portion of the grids can be damaged and are not really usable for electron microscopy. So the chameleon is a new approach. We try to address all these issues by taking a new route. Here we are speaking of a fast, lossless approach to try to prevent or to limit at least the interaction at the air-water interface. And this is based on two particular features of the chameleon. First, the chameleon is using a nano dispenser to spray the sample on the grid, and it will project small droplets of your sample on the surface of the grid. And the grid is a specific grid, which is a set-working grid, a quantifiable grid covered with this small nanowire made of copper. And by capillarity, this nanowire will absorb the droplets, so there is no need to come with a blotting paper to take out the liquid. The grid itself will remove the droplets from the carbon support and make a thin layer of ice upon vitrification. So it's the combination of these two approaches that makes it possible to remove the needs for blotting paper. And by removing the blotting step, actually, you can make a fast freezing. Fast freezing means that between the time that you spray your sample on the grid and the time that the sample is vitrified, as little as 15 milliseconds has happened. You can, of course, do longer wicking time if required, and you can go up to 2 seconds, depending on how the sample behaves and its concentration. If you take a zoom on the chameleon device, the platform on top looks like this. And the very nice features of this system is that it's fully automated. So you limit to the maximum the manual handling of the grids. The only steps which require human intervention is to deposit the grids in this red platform. And then after that, the robot take over. This tweezer will come, pick up your grids, bring into the glow discharge unit, which is integrated into the system. After glow discharging, the grids will be unloaded and presented in this rod to the nano dispenser. The nano dispenser is located in this part, is also automatized, and so it will aspirate your sample from a vial which is located here, which is temperature control to keep the sample at the desired temperature. And after aspiration, the nano dispenser will go to the shroud also to spray the sample on the grid. After spraying, the tweezer will plunge to cryogen, which is located below this black part here. So the lid will open and deposit the grid in the liquid ethane. The nice feature also is that you can have direct feedback of the session from a dedicated camera located in the shroud and taking images of a grid while it's being sprayed by the dispenser and just right before plunging. So you have an opportunity here to assess the quality of what you have been doing and to also, if you want, reject or accept the grid and avoid to look completely dry grids or black grids where the ice is just too thick and are not really usable for microscopy. Finally, use also very small amount of sample. It's true that you don't need so much with a Vitrobot, but here it's even better because with 3 microliter, which is a minimal volume that you will be able to put into the chameleon, you will be able to perform more than enough grids for your sample. Not perfect, of course, and there are still limitations. And the major one for me, at least, is that it requires a high concentration of sample compared to the Vitrobot, up to 10 times what you will normally use for plunge freezing. And it's true that for some projects where the sample is difficult to produce, it can be a limiting factor for using the chameleon. It's also quite sensitive to sample quality as it's based on the spraying of very small droplets from the dispenser. And so if your proteins have a tendency to form aggregates or form big structure like fibers, for example, this can clog the dispenser and result in very inconsistent dispensing of the sample. And so this might be difficult to reproduce or to obtain good grids. For the very same reason, it's also sensitive to viscous buffer, even though there is no hard recombination compared to what you will have for the robots. But detergent or glycerol or really viscous buffer could be problematic for the dispenser. Okay, that looks nice on paper, but does it work actually? I'm sure you have this question. And the short answer is yes, it works. For some cases, we have seen improved stability of the sample. If you compare, for example, in this particular case, the VitroBot to the chameleon, the user have spotted that a lot of aggregation was visible with the VitroBot and could not be observed with the chameleon. So we have improvement of sample quality under the microscope. In this example, actually, the major limitation was the disruption of the interaction between the protein and the DNA that was studied. And using the chameleon, the user could dramatically enrich in the proportion of sample of protein still binding to the DNA, which even led to the first reconstruction of the complex sitting on the DNA. And also a third example, actually, where using a plunge fusing approach, again, the stability was improved. A lot of aggregation was visible on the grids, or with the user was seeing only small objects on the grids. And where we're using the chameleon, bigger particles were visible in the images, and that was really clear using a 2D classification actually. But with the chameleon, they mostly had intact complexes, whereas when using the Gatan plunger this time, there was a lot of complex dissociation. So, this conclusion about the chameleon, does it address the limits that I presented to you of the blotting approach? Well, it seems to answer some of the questions at least, and the air-water interface problem, while it's not completely eliminated, it's still working a bit better with the chameleon, and for some cases it's beneficial. And what we have seen so far, at least in the case that we have studied, that it's by improving the stability of the complex on the grid. You have the opportunity also to reject grids, which looks obviously bad on the camera. You still need to check your grids for particle distribution and quality. Of course, this is not something you can avoid, but it can already increase the throughput of the screening part, because you reject immediately the bad grids. You really don't need a lot of material, as I said. You need higher concentration, and that can be limiting. But also now there are protocols to produce graphene-covered grids for the chameleon. We have not tested it yet. This is something to try in the near future, and this can be a solution to this particular problem when the sample is difficult to produce. Automation is a very nice feature, and indeed it's much easier to handle once you know how to use it, and there is much less damage and grids which are rejected because of bad handling of the grid from the user. Just to finish, unfortunately, it's still not a silver bullet, but we have other solutions to address very difficult sample. We propose streptavidin affinity grids, but I guess this will be the topic of the next presentation. With that, I would thank all the people involved in the cryo facility, the user who gave me the opportunity to present the results, and SPT LabTech crew for their support when running the machine. And I'm happy to take any questions.
Thanks a lot, Alexandre, for a great presentation. So we have a question from Pedro Matias that is asking, why did you choose the chameleon over the VitroJet?
So the principle seems new to me when you check the chameleon compared to what the VitroJet is doing. So it's difficult to really make a clear separation of the two methods without checking some samples and getting some results out of it. The idea that fast freezing could be beneficial to a sample preparation for me was bringing something new. And from what we could test, actually was working. So this was the main point on taking the chameleon rather than the VitroJet.
Thanks. And I will ask a very quick question. So in your pipeline at the moment, do you always use the VitroBot first And if you have issues with the samples, you go towards Chameleon, or in some cases, you know that the sample would fit better with like the Chameleon technique for sample preparation.
So far, we always test the VisRobot first, and we go to the Chameleon for difficult sample when we clearly see problems with the vitrification method. So it's more a solution, but something that we propose generally. And that's because first, it's not, I mean, as I said, you can be limited by the protein concentration. And also because, generally speaking, it's easier to perform the VisRobot experiments, and the user independent to do that. So it's more integrated into the workflow. Also, you have less possible data collection area on the grid because you spray a single band. So it might be tricky to find enough region for data acquisition. So this is not something I will use as a first attempt when running a new project.
And we have a last question from Hugo. With Chameleon, what are your favorite grids for 300 angstrom molecules?
So with Chameleon, what is my favorite grid?
Yes.
What do you mean by this?
Maybe we can ask Hugo to ask the question directly if he raises his hand. Yes. Hugo, you should be allowed to talk now.
Hello.
Yeah.
Hear me?
Yeah.
Yes, what I mean is what type of grids do you like or do you prefer? What are your favorite grids? Do you use copper grids? Do you use gold grids, ultrafall?
At the moment, it's not like we have a choice because the only available grids are copper grids with carbon film. I know that gold grids will be available soon, or at least they will be. But according to SPT Labtech, they should be available soon. We already had one user with the chance to test them, and it looks promising. So for now, the only one that we have are the carbon and copper, and we hope that the gold grids also will be available soon for more regular usage. Because it's true that for cryo-EM, generally speaking, the gold grids are working very nicely and working better.
Mm-hmm. Thanks a lot.
Thanks, Alexandre. So I would like to thank once again all the speakers for this great webinar. If you have any question to our speakers, don't hesitate to contact them. They are all part of Instruct, so you can find them on our website easily. And I wish to see you all for our upcoming webinar that will take place in June. And thank you all for your attention. Bye.